Previous video, we have seen how the genetic information present in the DNA of a cell is transcribed to form the RNA, the messenger RNA. Now, this messenger RNA, which is a sequence of ribonucleotides, it codes for the various amino acids that will form the ultimate product, that is the protein. So in this video we are going to see how this RNA molecule, it leads to the formation of a sequence of amino acids, that is a polynucleotide sequence leads to the formation of a polypeptide sequence by this process of translation. Now, certain basic requirements for this process, they include, first of all, the messenger RNA, which has got these nucleotides, the ribonucleotides, and we know that this ribonucleotides which form the messenger RNA, they have got the 5' cap and a poly A tail. Apart from that, it has got certain untranslated regions which do not code for any amino acid. They are required for the attachment of the machinery that will lead to the reading of these nucleotide frames. And such untranslated regions, they are present both on the 5' part of the coding sequence as well as on the 3' part. The cap part, as we know, it is the inverted G guanyl nucleotide, and the polyadenyl tail is present on the 3' end. Now, we know that the coding region, it starts with the start site and at the end is the stop site. Now these start site and the stop site, they basically are defined by the codons that initiate the process of translation and ultimately are they indicate the termination of this process of translation. Another well-known fact that we are well versed with is that the sequence of 3 consecutive nucleotides present on a messenger RNA, they code for a single amino acid. They are basically the 20 essential amino acids, and a triplet code is specific for each amino acid. So in this manner, the sequence of nucleotides present in the coding region, they define the sequence of amino acids that will form the ultimate product, that is the protein. So this messenger RNA, it is the basic requirement of the process of translation, because we are dealing with the translation of RNA. And this process of translation, it is supported by or assisted by other molecules. There's another type of RNA known as the transfer RNA, because messenger RNA, it has got the triplet codon, each specifying a particular amino acid. So we need various amino acids and some adapter molecule which can recognize the messenger RNA, a particular codon, and along with it, it may carry the particular specific amino acid. So, such type of adapter molecule is the transfer RNA molecule. It was Crick who gave this concept of adapter molecule, the molecule which co-links the process of reading of these codons and attaching the specific amino acid in the sequence as defined by the nucleotide sequence of the messenger RNA. So we have done about the detailed structure of this transfer RNA in another video that deals with various types of RNAs. So here, this transfer RNA, it brings along an amino acid. Now we know the amino acid, it is basically an organic molecule having the amino group on one end and the carboxyl group on another end. And the various amino acids, they differ in the side chains present in between the two ends, that is the N and the C end of this amino acid molecule. Now when these amino acids, they are brought about on this uh, coding sequence, on this genetic code, the two adjoining amino acids, they form the peptide bond. We say that proteins, they are polypeptide molecules. Molecules in which various amino acids, they are connected to each other by means of peptide bonds. So for the formation of this peptide bonds, the hydroxyl group of this carboxyl, the OH part of this carboxyl group, it reacts with the H, it combines with the H of the amino group of the next amino acid, and this leads to the formation of a molecule of water, and a bond is formed, that is a peptide bond is formed between these two amino acids. So as this chain will increase, the protein, it will have an N end and a C end. The next amino acid that will come, it will use this hydroxyl group and the H of the incoming amino acid. So in this manner, the various amino acids, they join with each other, they connect with each other, resulting in the formation of protein. Now we have got the messenger RNA. This messenger RNA has got the genetic code, which is read by the complementary anticodon uh, present on the transfer RNA molecule, which is bringing along the specific amino acid. And these specific amino acids, when they come to lie together, they form the peptide bond, resulting in the formation of the protein molecule. But all this complex phenomena that takes place, it has to have a certain machinery so that the position of these various RNAs, it is fixed, so that site of synthesis of this polypeptide chain, it is provided by the organelle known as the ribosome. Although we have done learnt about this ribosome in another video, but here we may just revise that this organelle is a membraneless organelle. It does not have a unit membrane enclosing it, so it is present in both prokaryotic cell as well as eukaryotic cells. It is made up of two subunits, the large subunit and the small subunit. Each subunit is basically made up of different types of ribosomal RNA. Now, this is the third major type of RNA present in any cell. The messenger RNA we have done, the transfer RNA we have done, and now it is the ribosomal RNA, which along with various proteins, they form these structures, that is the large subunit and the small subunit. These ribosomes, they are present in both prokaryotic and eukaryotic cells, but they are of different types depending upon the mass. In eukaryotes, the large subunit is made up of 28S, 5.8S, 5S ribosomal RNAs along with 50 different types of proteins, and these together they form the 60S subunit. Now here S, it is the Sedberg unit, which is a measure of the mass of this part of ribosome. This large subunit, in case of prokaryotes, it is made up of two types of ribosomal RNAs, that is the 23S ribosomal RNA and the 5S ribosomal RNA, and along with 34 different types of proteins, and its mass is 50S. Similarly, the 40S subunit or the small subunit of a eukaryotic ribosome, it is made up of 18S ribosomal RNA along with 33 different types of proteins. See, it is a very complex organelle that is made up of so many different types of ribosomal RNAs and so many different types of proteins, because what we are seeing as a simple phenomena in this video, gradually in the upcoming videos we'll deal with the details of various uh, subunits and the various proteins and the functions. So in prokaryotes, the small subunit, it is made up of 16S ribosomal RNA and only 21 different types of proteins. So these two subunits, they join together, they fix this messenger RNA, and they give a space to the transfer RNA so that the different amino acids, they may join together to form the polypeptide chain. Now we'll try to have an inculcate an understanding as to how this polypeptide chain, it is formed using these basic um, ingredients or the basic raw material. One more thing that is required apart from these various amino acids and the various types of RNAs is the enzymes, the different types of regulatory proteins which regulate the three phases of translation, that is the initiation factor, the elongation factors, and the release factors. So the machinery that is required for the process of translation, it is quite complex, and so is the process. However, simply speaking, this uh, process of translation, that is forming the protein with the help of RNA, it can be divided into three basic steps. first step is the initiation step. Now, during the initiation step, the messenger RNA, it connects itself or it joins to the small subunit of the ribosome, and the 5' untranslatable region is the site where initially this small subunit it attaches itself. Gradually it moves to the start codon, that is the AUG, and in this ribosome there are three specific sites: E, P, and A. So these three sites, they are present in the small subunit and the corresponding larger subunit. So this P site of the small subunit, it comes to lie on the uh, initiation codon, the start codon, that is the AUG. Once this happens, the transfer RNA, which is carrying the specific amino acid to initiate the process of protein synthesis or translation, it has got this anticodon for the AUG codon, so it will come and attach to the start codon. As we have already said, that each transfer RNA it carries a specific amino acid, and the transfer RNA that recognizes this AUG, that is the start codon, it brings along methionine. But this methionine, it is the formilated methionine. That is, a formyl group is added upon this methionine, which forms the which comes and initiates the process of translation. Now, once this setup is ready, that is the small subunit attached to the messenger RNA and the specific anticodon present on the transfer RNA carrying the amino acid, it attaches to its respective codon. When this happens, the large subunit it attaches with the smaller subunit, and now this initiation complex is ready. So formation of this initiation complex, it involves various initiation factors about which we will learn in detail in the upcoming videos. So once this formilated methionine carrying transfer RNA is present on the P site, next phase of uh, translation it takes place, it initiates, and that is the elongation phase. Now the next triplet code is in this A region, the amino acid region of this ribosome. So the next transfer RNA which carries the respective amino acid, it comes and occupies this point, that is the A site of the ribosome. Then this uh, P site, the transfer RNA present on the P site, it transfers its amino acid to this, and a peptide bond is formed. Now this transfer RNA, since it has given its amino acid to the adjoining transfer RNA, it is free, so it shifts its position to the E point. and gradually, subsequently, the transfer RNA, which now has got these both amino acids, it comes to occupy the P site. Now this shifting of sites is because this ribosomal machinery, it moves along this messenger RNA from the 5 prime to the 3 prime direction, in the 5 prime to 3 prime direction. Now it is the same direction in which the transcription takes place, in which the messenger RNA is synthesized. It is because in case of prokaryotes, we know that the site of transcription as well as translation is the cytoplasm itself. So it is not that the process of translation, it waits for the entire messenger RNA to form, and then only it will start the formation of the um, protein molecule. No, it's not so. In case of prokaryotes, as soon as a stretch of messenger RNA is available, that is, it has been transcribed, the process of translation, it initiates. Now the transcription machinery, it is moving, and along with it the translation machinery, it has also initiated on the part of on the part of RNA that is synthesized or that has already been transcribed. One more characteristic feature of protein synthesis is that on a messenger RNA, multiple ribosomes, they may mm, attach or they may start the translation process. That is, as soon as a ribosome moves ahead, the 5 prime region or the 5 prime UTR is made available for the second ribosome to attach itself. So multiple copies of protein, they are formed on a single messenger RNA, and the messenger RNA with multiple ribosomes attached to it, it is known as the polysome. So since this ribosomal machinery is moving ahead from the 5 prime to the 3 prime region, these triplet codes, they change their position within this ribosome, and the nucleotides which previously were present at the P site, they are now present on the E site, and those which were present on the amino acid side, they come to this peptide formation, this P site of the ribosome. And in this manner, now again this A site is available for the next transfer RNA to come and attach itself. So gradually this chain of amino acid, it increases, and the ribosome, it moves ahead during the elongation phase. Here again various elongation factors or various proteins, they are involved in this process, and the details about these elongation factors and what actually happens, that we will deal with in another video in detail. So in this manner, this ribosome machinery, it moves ahead on the messenger RNA, and this polypeptide chain of various amino acids, it is synthesized or it is formed. This process, it takes place until and unless on this A site the stop codon, the UAG or the UAA or UGA, they come across. And since there's no transfer RNA which recognize these particular genetic codes, which we call as the stop codons. So no transfer RNA will come to occupy this point, that is the A site, and instead some protein, some release factor, it will take this point, it will occupy this A site, and it will release the transfer RNA present on the P site. So this amino acid chain, it will be released. That is the free polypeptide chain we will get, the messenger RNA we will get, and this release factor, it also separates the two subunits of the ribosome, that is the smaller subunit and the larger subunit. So in this manner, the translation of ribosome of RNA takes place with the help of ribosome. This is the site of translation, and it leads to the formation of the final product, that is the free polypeptide. So the in the upcoming videos we will deal with the three uh, processes of this translation phenomena, that is the initiation, the elongation, and the termination part, when we will see the details of the various initiation factors and the elongation factors and the release factors that are involved.